आज क्या स्टार्ट करने वाले हैं हम? मनप्रीता,
सर लास्ट क्लास में एक्टिनोमाइसीट्स हुआ था।
okay. That means we have already discussed eubacteria, then cyanobacteria, mycoplasma, archaebacteria, okay, and actinomycetes also. Okay. So we are to start protista today. So remember, protista, it's a group, it's also known as, it's an artificial kingdom under R.H. Whitaker system of classification. Why it is so? Because what is the definition of protista? Protista includes all the unicellular and eukaryotic organisms. Is this clear? So it is called an artificial kingdom. Why? Because kingdoms, members from other kingdoms are also kept under protista. Okay. And because of which the strict rules and regulations are not being followed. Okay, so what is happening here? You'll find निकुंगनी, नंदिता, मनपिता, डेविड और कौन था? So it is an artificial kingdom under RH Whitaker system of classification as members of all other kingdoms are arbitrarily kept under this kingdom if they are unicellular and eukaryotic. Okay, without maintaining proper phylogeny. Okay, so that is the reason why it's called an artificial kingdom under RH Whitaker system of classification. I think you can remember. Next point, what you can say? Uh, it is a connecting link between Monera and all other higher kingdoms. So protista is called a connecting link. Okay, protista itself called a connecting link, and within protista specifically it is slime molds. Okay, so remember this. Now characters, general characters, if we see, then you can find they have all the general properties of an eukaryotic cell. Clear? So you can find 9+2 arrangement of flagella. In the flagella, you'll find 9+2 arrangement of microtubule is there. Then all the an eukaryotic cell have all the organelles, membrane-bound organelles you can find here. But here, certain points like a cell wall, okay, cell wall may be present, or absent. Okay. So most of the characters will be like this. Motility may be present, may be absent. Cilia may be present, may be absent. Pseudopodia may be present, may be absent. Food in nutrition, you may find autotrophic, autotrophic, heterotrophic, as well as you'll find mixotrophic. That means what? It is showing both autotrophic as well as heterotrophic mode of nutrition. So that is called mixotrophic. So, for example, Evelina. Evelina is what? Autotrophic or what? Evelina is autotrophic or not? Manpita, Evelina is autotrophic or not?
Sir, both.
Autotrophic and what?
And heterotrophic.
In heterotrophic, which mode it shows?
Sir, what?
In heterotrophic, what is the mode of nutrition?
Sir,
Symbiotic, parasitic, holozoic, saprophytic, what?
saprophytic.
Huh?
Saprophytic.
Saprophytic. Exactly. Remember, so they are autotrophic plus they may be saprophytic. That means you cannot find holozoic mode. Acha. So remember this. So you can find all these types of properties are there. That's why I'm not writing any other properties. Simply, if you find the properties of protista, you can say what? May be present, may be absent. Now tell me 9+2 arrangement of microtubule is present or absent? 9+2 arrangement of microtubule is present or absent?
Present.
Yes, present. Definitely present because it is eukaryotic. So membrane bound organelle is 70s or 80s ribosome. Which one is present?
80s.
No. Both are present. In cytoplasm, if it is asked that 70s or 80s, which one is present? In cytoplasm, which one is present? 80s ribosome is present. In mitochondria and plastid, 70s is present, is not it? So both 70s and 80s are present. So if specifically asked that in cytoplasm what is present, then you write 80s. Otherwise, what you say? Both 70s and 80s are present. Is this clear?
Yes, sir. Yes, sir.
Okay. Then protista, you'll find protista we have categorized as plant-like protists, animal-like protists, and consumer decomposers. Plant-like protist means what? These are autotrophic. Okay. Then you can find consumer decomposers. Consumer decomposer and what? Animal like protists. Now tell me just one thing. If it is animal like protist, then it can be further classified into what? It may be flagellates, or you can say it may be amoeboid, is it clear? It may be amoeboid, it may be flagellate, it may be ciliate, or it may not have any locomotory organ. Then these are called Sporozoans. Can you tell me the examples? Amoeboid example is what? Amoeba, entamoeba, flagellate example. Who knows?
Goniolax.
Oi,
and
these are
then
animal-like protists. Flagellate.
Euglena.
Euglena is plant-like protist. Animal-like protist we are discussing. Hmm? Have you heard about trypanosoma?
Yes, sir.
What is that? Causes sleeping sickness. Does it have flagella? Trypanosoma causes what? Sleeping sickness. So remember this, very important. Ciliate, paramecium, have you heard? Hmm. So paramecium is ciliate. Sporozoans. Have you heard about malarial parasite? What is the name? Hmm?
Plasmodium.
Plasmodium.
Plasmodium filets.
So Vibex is the most potent. Altogether, 4 types of plasmodiums are there, no? So sporozoans proteins. That means what? They do not have any locomotory organ. When they get infected there inside our body, then they travel along with the bloodstream. Clear? So these are animal-like proteins. Consumer decomposer. That means what? These are nothing but slime molds. Okay, these are nothing but slime molds. So in slime mold, again, we can categorize as cellular, acellular. Okay, so many things. But simply, I'm not discussing because it is not given in NCERT. So remember, Phycerella, Phycerum, Fuligo, Dictydium, Dictyostelium. So many names are there. I'm giving just 2 examples. Try to remember. Phycerella is very important. Okay, slime mold. So remember, Phycerella, Phycerum, you remember. Plant-like protists, we can further categorize them into what? Into 3 categories. Diatoms, you can write dinoflagellates, and then you can write euglenoids. Is that clear? Dinoflagellates. So this plant-like protist we will discuss with more details. We are interested here. These are the food types. Slime molds, I'm not differentiating them. Diatoms, dinoflagellates, and euglenoids. Remember them. So If we classify them, we'll find that the different types of protista. Have you drawn this? Is it over? Then let us start with Dinoflagella. Dinoflagellate's main characters, if I draw a structure, we can say it has a body has two groups. Check on its body, you can see two distinct groups are here. One is longitudinal group, another is transverse group. Longitudinal group is known as what? What is the name? That longitudinal group is called sulcus, and this transverse group is called annulus or girdle. It is called annulus or girdle or singular. So many names. You'll find a structure, say two flagella are there. One is laterally inserted and small. Another is inserted in the longitudinal groove and it is a very long one. So having two different types of flagella, okay, is known as what? Heterofont condition. What is the name? Heterofont condition. Okay, remember this. Now, its whole body is covered by a thick, see, spectrocellularic structure. Okay, and that is called theca or lorica. Cell wall is called theca or lorica. And because of this theca and lorica, the organism look like uh, as an armored structure. So these organisms are also known as armored protist. write these points in Word document. One second. David, can you hear me?
Yes, sir.
Okay. Better we write Dino plus. Okay. Right. Can you see properly? So number 1 point you write, these are unicellular, motile, mostly marine forms, but some occur in fresh water. Okay. Cell wall is called lorica or theca. Made up of stiff cellulose plate. Theca has longitudinal groove. Longitudinal and transverse groove. Whereby longitudinal groove is longitudinal groove is called sulcus. Okay. Transverse groove is called annulus. Or cingulum or girdle. Okay. Heterocon flagella you have written.
Yes, sir.
Orientation flagella. What you can write? Both the flagella beat at 90 degree at a right angle, right angle, angles, producing a spinning movement, which is known Whirling whip. W-H-I-R-L-I-N-G. Whirling whip. W-H-I-S. Okay. Remember this term. Next, you can say photosynthetic pigments. Okay. So photosynthetic pigment is not given in NCERT. If you can remember, it's very good because sometimes questions are asked. Okay. Not directly based on similarity, they may ask you question, but if you do not know at all that what is the photosynthetic pigment, then you cannot answer. So better to remember this. Photosynthetic pigments. Chlorophyll A and chlorophyll C, along with which you can find alpha carotene. Alpha carotene and xanthophyll. Okay, so these are the photosynthetic pigments. Reserve food is basically carbohydrates and oil. Then you'll find a specific vacuole that is called actually non-contact. It is aquatic, marine. No? So marine organisms do not have any contractile vacuole. Always remember, marine organisms cannot have any contractile vacuole. There is no utility of any contractile vacuole. So you'll find a non-contractile vacuole called pusul. Okay, pusul is present, which help in floating and osmoregulation. Then come to reproduction. Asexual reproduction through cell division, you can see, and cyst formation, right? Sexual reproduction can write it generally absent, reported in some members by anisogamy or by isogamy or red tide. We have discussed it the last class also. Is caused by whom? Gonyulex. Gonyau. Gonyulex bacterium. It produces saxitoxin. Have you heard this term PSP? Paralytic shellfish poisoning. Bigonulates. Oh, acha. Bigonulates. Okay. It will be Gonulax, gymnodinium also you can read gymnodinium because sometime in uh, some question paper I have seen they don't give Gonulax alone, along with which they often add gymnodinium. Gymnodinium also possess red tide. Okay, what is red tide? That means this poisonous this one dinoflagellates, if they grow profusely in water, then the color of the ocean becomes red. Huge area becomes red. Okay, so that is called red tide. Let me show you. Can you see this color? Hello.
Yes, sir.
Yes, sir.
Check it here. All the fishes are dying. All the aquatic organisms they die. Why they die? Because of this poison. Sometimes what happens? Let's say all these fishes, marine fishes have died. So this is because of red tide. See the color becomes complete red. Okay, check it. So this red tide is very dangerous actually, in contrast to red sea. So in red tide, poison, see, so many fishes are dying. Is that clear? So these are the causes of red tide. See, Chile's worst ever red tide kills 20 million fish. So this is the condition of red tide. So this red tide is very dangerous and it is caused by Noctiluca. Some of the, oh sorry, this is caused by this Conilus. Another member you should remember Noctiluca. Take it here. Can you see? This sea water you see, the color of the water you see. Can you see? If anybody touches that part of the water will glow. Why the glow? Because here some of the this dinoflagellates which are bioluminescent, okay, which are bioluminescent, they are growing, and that's why what you find the part where Noctiluca or some other say Pyrocystis, Pyridinium, some other uh, members are there. When they grow, then they glow at night. Beautifully, they glow at night. So these are Noctiluca. Can you remember Noctiluca? Remember Noctiluca for many things. Noctiluca is the only diploid member and it is bioluminescent. Okay, so you have to remember Noctiluca. So write about them. So this much points are sufficient for this group. Next is chrysophytes. Actually, these dinoflagellates, they belong to pyrophyta, remember. Pyrophyta. Okay. They belong to pyrophyta. And under chrysophytes, actually two types of members are there. One is diatoms and another is desmids. So both come under chrysophytes. So under chrysophytes, directly, because in syllabus only diatom is given, we can directly write diatoms. There is no issue. So it includes, this group is called actually golden algae. Okay, these desmids are also known as golden algae. So you just write, this group includes diatoms and desmids, which are also known as golden algae. Next, you write the causes, their sewage pollution indicator. Remember, interesting point. Non-flagellate. Okay. Non-flagellate, that's a very important point. Flagella may appear at the time of reproduction. Okay. Or better right, appear in a reproductive stage. So if flagella is absent, so how they move? They float passively in water current, okay, with the help of or due to the presence of lightweight lipid and a large vacuole within the cell. Now, their cell walls are very important. The cell walls are very important, remember. Silicified cell wall, known as crustule. Cell wall is indestructible due to the presence of silica. It forms diatomite or diatomaceous art, which is also known as K cell good. K-I-K-I-E. G-U-H-R or R-H-H. Good. Yes. K cell good. Now see this K cell good. So what happens? This diatom, when they die, their shell, that cell wall or the frustule, they gets deposited. So when this shell gets deposited, then this shell forms what? This shell forms a thick covering actually, and this shell is very useful uh, economically. So this shell actually ultimately gets converted into diatomite. It is called diatomite or diatomaceous earth or kisel gold, locally known as kisel gold. So this kisel gold, it is gritty in nature. Okay, you can write because of gritty nature. It is used in polishing, filtration, filtration of oils and syrup, mm, in cosmetics, like in the preparation of scrub, we can produce, in the preparation of dynamite, in the preparation of toothpaste, okay, in the preparation of toothpaste, in the preparation of soundproof brick, okay. So these are used. These are used. So remember, I'm not writing all the points, just remember. Cell wall consists of, actually they have a soapbox like cell, okay, or cell wall. Cell wall consists of two overlapping shells. That is epithecia and hypotheca, which fit together as in a soap box. I think this is clear. Photosynthetic pigments include chlorophyll A and C. Okay, along with which they have beta carotene. Dia, diatoxanthine and diadenoxanthine. Remember these pigments? See, they have chlorophyll A, C. In earlier case also in diatoms we have found, the dinoflagellates also we have found what? Chlorophyll A, C. But there it was alpha carotene, here it is beta carotene. Beta carotene is also found in higher plants. Okay, so there is slight difference. Diatoxanthine and diadenoxanthine are special xanthophylls which are present in dinoflagellates. Remember, very important. Reserve food material. You can write it is very simple. It is oil in some cases, or we can say what we find in pheophysi, chrysolaminarin, means laminarin and mannitol we find no, there. So here it is chrysolaminarin because of this group. Chrysolaminarin, it's a bit different type of laminarin specific to this group, and leucosin. Next, you can write asexual reproduction by mainly by binary fission. And here, very interesting phenomenon occurs. I'll discuss now sexual reproduction occur by gametic fusion, by isogamy, anisogamy and oogamy. It has diplomatic life cycle and gametic meiosis. Remember this class. Here, one very interesting phenomenon. Check, this is a so box-like structure, right? We'll find this organism has a structure like this. So one fits on another. We'll find the structure is like this. So what happens when division occurs? Then what happens? The two contents, the inner side is containing the organism. Let me go ahead. See, this is the organism. This is the cytoplasmic content. You will find it has a nucleus. It's a nucleus. It is like this here. And one very interesting fact is its cytoplasm remains connected to the cell wall only at certain point. And in rest portion, a big vacuole occupies. And because of this, it forms a structure like this. This is cell wall, and this green part, it is also, it is what? It is plasma membrane. And because of presence of big vacuole, cytoplasmic content will be present here. So I can say this much is the cytoplasmic content. Okay, this gap is what? This gap is a big vacuole. So you can mark this part. This gap is called vacuole. This vacuole helps in floating, plus they have what? There's a lightweight lipid. Okay, lightweight lipids are also there. Say one diatom is dividing. So its two structure, epitheca and hypotheca, they get separated. So when they get separated, they will be separated like this. So tell me now, the portion which has received epitheca will develop what? Manpita, Nikumani, the portion which has received epitheca will develop what? Hypotheca or not? Hello. Hmm. Can you hear my voice?
Yes, sir.
Then why are you so silent? कुछ बताओ, किसको क्या मिला है बोलो तो? What is the name of this structure? This is called epitheca. तुम लोगों को समझ में आ रहा है ना? This is called epitheca. কোৱা কোৱা নিকুমণি। So it will develop hypotheca or not?
Yes.
Okay. It's an important point. It's অলপ ভালকে বুজিব লাগে। So if it has got epitheca, it will develop hypotheca. So consider this is hypotheca. It has got its hypotheca, right? Now the cell which has received hypotheca will produce what? Epitheca or hypotheca?
Epitheca.
No, it cannot produce any epitheca. It will produce every time it will produce a hypotheca. Check. It has got hypotheca, so it will develop another hypotheca only. It cannot develop epitheca. Is this clear? So every time what will happen? If it synthesizes a hypotheca, so automatically out of these two cells, check it this, out of these two cells, one will be smaller and another will retain its original size. Is this clear? Hmm? So out of
Yes.
these two cells, one
Yes.
is smaller. Now, again, if this cell divide and it is receiving hypotheca, so this hypotheca will generate another hypotheca. So after few division, what will happen? The size of the cell will become even smaller, right? So after few division, the size of the daughter cell becomes so small that they cannot go further division. So under that condition, this structure, the inner content, they come out of the spustule. Can you remember? This inner content, the inner content, say this is the inner content. The inner content come out of the pustule. So when they come out of the pustule, we call them as a protoplast. Acha, one very important point, all these dinoflagellates are diploid. Can you remember? All the dinoflagellates are diploid. So when this inner content comes out, it will undergo meiosis. Remember this point. It will undergo meiosis. After meiosis, it will develop what? Large number of haploid cells. Are you getting my point? When it will produce large number of haploid cells, that means here it will be N, 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 N. Is that clear? So now any of these two cells, maybe between these two or maybe amongst other also, they will fuse and develop what? A diploid condition. What is the name of that condition? That is called a zygote. Is this clear? This is a zygote. Now, this zygote, very interesting. This zygote will now grow in size. Okay. So when zygote grow in size, it will regain its original shape. Okay. So zygote will grow in size and regains its original shape. When zygote grow in size, then it is known as, that spore is known as oxospore. So this is zygote. These are all gametes. Gametes are produced after fertilization. This is fertilization, you can say. After fertilization, what will be produced? What is this? Zygote. So when zygote grow in size, that cell is called oxospore. Okay, that cell is called oxospore. Now, oxospore, it will develop pustule. Is this clear? Draw this card. Give me 1 sec. I'm coming. Is it visible? Give me 1 second. I'm coming.
স্যার আপুনি মিউট হৈছে।
Is it done?
Yes, sir.
Okay. So up to this much is dinoflagellates. Next is eugenides. Where euglena is basically found.
Stagnant marine water.
Exactly, stagnant water. So you can write flagellate protist found in where? Stagnant, in fresh stagnant water or fresh and stagnant. Okay. Cell wall is present or absent? Remember, in euglena, cell wall is not found. Body is surrounded by an elastic and flexible proteinaceous structure. Okay. Called pellicle. Can you remember? Next, we'll find generally they have two flagella. Means the two flagella originate from different points, right? Two flagella originate from different points, but what happens? They unite at a particular point. Let me draw it. It will be very much clear. If you see Iglina, say this is there is a cytopharynx. Okay, so there is a cytopharynx, and what happens here? We'll find one flagella. There is another flagella. The shorter flagella, it fuses with the longer flagella at this point. Okay, and that means they have two independent basal granule from which they have independently originated. But at a certain point, they will get fused together, and the point where they fuse together, it is photosensitive. It can detect light. Just parallel to this, there is another red colored portion that is called eye spot or stigma. It contains a pigment that is photosensitive pigment that is known as astaxanthin. Can you remember? This is called astaxanthin. This is called paraflagellar body. Okay, you write it down. Astaxanthin, and this is paraflagellar body. This whole thing is called cytopharynx, okay, or cell mouth. Having this type of flagella is called what? There's a specific term that's called stichonomatic flagellum. Okay, that means one flagella having two basal bodies are called stichonomatic flagella. So write some points. They have two flagellae, a short and a long. Heterocon flagella. Longer flagellum is stichonomatic. Stichonomatic. Oh, they they have two flagella. Two flagellum. They have they have two flagella, a short and a long. Heterocon flagella. The longer flagellum is called stichonomatic. Stichonomatic having a row of hairs called masticonims only on one side. Evelina has mixotrophic nutrition that you know. There is photosynthetic, synthetic and heterotrophic. When deprived of sunlight, other euglenoids behave as predators on smaller organisms. Okay. Photosynthetic pigments, it is just like for our higher pigments, higher plants. Photosynthetic pigments are resemble higher plants. Can you tell me which photosynthetic pigments are found? Chlorophyll, A and B. Then beta carotenoid, we have already written here. Beta carotene and xanthophyll. Reserve food is what? Paramylum or paramylon. Paramylon is also known as beta 1,3, beta 1,3 glucan. So I have written reserve food. Contractile vehicle is present in not in euglenoids.
No, sir.
Why not? It is marine or freshwater. In marine form, contractile vacuole is not found, but in freshwater form, you will definitely find a contractile vacuole. Why? Basically for osmoregulation. Helps in osmoregulation and excretion also you can write. Next you say, what is the reproduction method? Asexual reproduction takes place by longitudinal binary fission. Very important point. Many times this has been asked. longitudinal binary fission. Okay, sexual reproduction is not reported. And that's why you do not know whether they are haploid or diploid. Evelina is connecting link between
Sir, animal or plant?
exactly plants and animals. Bas. So this much is for Evelina. It's very simple. Not much point I have added. Only the most highlighted points I have added here so that you can easily remember. If you can remember this much, that is more than sufficient.
Yes, sir.
Acha, paramecium. In paramecium, how many nucleus is present? You know, as a note you write. Earlier, many times this question has been asked. In paramecium, two nuclei are present. The larger one take part in metabolic activity and the two nuclei are present. The larger one take part in metabolic activity and the smaller one is smaller one take part in reproduction. Bas. So this is a note. You remember just this point. So next group is which one? Saprophytic protistans. Okay, and these are also known as what? Consumer decomposers. Consumer. So what is consumer decomposers? Next time, number one, right? They are commonly called as protistan fungi. or consumer decomposers. Naked protoplasmic body without cell wall is present where in the vegetative structure. Okay, and their spores contains cell wall. Let me draw the diagram, then it will be easier for you to understand. You will find in case of consumer decomposer, say check it here. Say this is an amoeba. Which one? Not amoeba. This is plasmodium. Plasmodium, not Plasmodium vivax. Okay. So here the slime mold it is forming in plasmodium. In this case, you will find large number of these are what haploid cells or no. There are two types. In one case, large number of haploid cells are present. So and another large number of diploid cells are present and they form plasmodium. Is this clear? So each and every cell let's check it. So this is a plasmodium. In unfavorable condition, we find, see normally it is an amoeboid structure. It develops pseudopodia and it may grow up to 2 to 3 meter. Okay, it may grow up to 2 to 3 meter. So what happens in unfavorable condition? Un, in unfavorable condition, they form some fruiting bodies. Is this clear? They form some fruiting body like this. And this fruiting body have cellulosic wall. Large number of fruiting body they will develop and the fruiting body also have cellulosic wall. Now they will undergo meiosis and develop haploid spores. Is this clear? So haploid spores develop. Now what you find from these haploid spore, they may develop in two different ways. They may form mexamoeba or they may form swarm cells like this. Motile cells, so these are called swarm cells. Afterwards, it will form a zygote. If the swarm cells are joining, then they will also form a zygote. That means it depends on the species. Some of them, they form swarm cells. Some of them, they form mexamoeba. So it will form a zygote. Consider this is a zygote. This zygote now divide repeatedly and will form this plasmodium again. Is that clear? So this is plasmodium. These are fruiting bodies. These are fruiting body. These are spores. And the spores also have cellulosic wall. So you can write spores also have cellulosic wall. So it will look like this. Okay. Spores are haploid. Spores on germination, they may develop swarm cells, or they may develop myxamebi. Okay, myxamebi. So they may either form myxamebi or swarm cells. Two swarm cells of opposite strain or two myxamebi of opposite strain, they may fuse to form zygote, or they may say undergo fertilization to form zygote. After zygote divide repeatedly, only karyokinesis takes place. So only karyokinesis takes place and develops this plasmodium. Is this clear? So in unfavorable condition, they forms a fruiting body within which meiosis takes place and spores develop. So this is the life cycle of acellular slime mold. This is the life cycle of acellular slime mold. Try to remember this. Is this clear? There may be another type that is called cellular slime mold. If it's a cellular slime mold, then what will happen? You'll find in that case, we find only single cells, and these are all haploid cells. In case of cellular slime mold, all these haploid cells, they live independently. So they will aggregate and form a plasmodium only in favorable condition. Only in unfavorable condition. So let me draw it in this way. Now what will happen? Inside, you can find lots of cells. They have aggregated to form the structure. But most peculiar thing is what? You can find division of labor. That means whole structure acts as a single organism. It's a cell aggregation, but whole structure acts as a single organism. So this structure is called pseudoplasmodium. Pseudoplasmodium, when it will develop fruiting body, it will develop only one fruiting body, not unlike earlier case, it will develop only one fruiting body, and the fruiting body is also not having any covering. So what will happen? Afterwards, spores will be germinated, spores will be formed, spores will be formed, and they do not form any swarm cells. They directly develop only myxamebi. Can you remember? So they germinate, spores germinate to form only myxamebi. Then these cells, they will unite together and form a structure that is called nuclear structure, which undergo meiosis, and afterwards we find it forms like this. Is that clear? So these are haploid structures. These are spores. The spores are, always remember, spore contain cellulosic ones. Okay, so these are spores. These are mixamoeba. Mixamoeba. Mixamoeba undergo fertilization and form a zygote. I think this is clear. This is the fruiting body. Remember, this is the fruiting body. This is the fruiting body. Okay. Okay then. Next in next class we'll continue this. In next class we'll continue this. This is cellular slime mold. Okay, this is cellular slime. Okay, thank you everybody.
Thank you, sir.
Okay, have you drawn this part?
Yes, sir.
Okay, that's good. So in the next class we'll continue this part again. Thank you.